The overall goal of this microscopy method is to precisely localize protein expression in relation to different subcellular organelles in the larval zebrafish retina by correlating super-resolution and scanning electron microscopy images.
This method can help answer key questions in the retinal development and ciliopathy fields, such as the study of protein mislocalization in ciliary mutants.
The main advantage of this technique is that it combines super-resolution with scanning electron microscopy to obtain highly accurate protein localization data within the native structural context of the sample.
The collection of Tokuyasu sections on silicon wafers substantially facilitates handling, and the use of platinum shadowing for contrast provides a good topography of the sample.
So this method can provide insight into larval zebrafish retina studies. One of its major advantages is that it can also be applied to many other biological systems, such as identification of protein expression in mouse tissue.
After dissecting the eyes from fixed zebrafish larvae according to the text protocol, warm up 15 milliliters of 12% local food brand gelatin in PBS at 40 degrees Celsius. Then, aspirate the PBS from the tubes of eyes and add the gelatin solution. Gently tap the tube to ensure the gelatin infiltrates the sample, and incubate it for 10 to 30 minutes at 40 degrees Celsius in a thermoblock with gentle shaking or in a water bath. In a 40 degrees Celsius water bath, fill 12 by 5 by 3 mm silicon or polyethylene flat embedding molds with warm gelatin.